A pleasant day for each and everyone. I am Jenaline Semando, presenting my thesis outline entitled Assessment of Plankton Abundance in Reference with Anthropogenic Activity in Nursery River in Dinapigi, Isabela. A plankton are a critical components of aquatic ecosystems, serving as primary producers and a food source of various marine organisms. Assessing plankton abundance can provide valuable insights into the health of aquatic environments, particularly in relation to pollution and anthropogenic activities. This assessment aims to investigate the abundance and diversity of plankton in the nursery river Dinapigi, Isabela, and analyze their correlation with pollution and human activities. The nursery river is one of the coastal town rivers of Dinapigi, Isabela, where community enjoy its natural biodiversity and state it has organisms present. However, most of the coastal citizens are not aware of their actions' implications to the water habitat. At the same time, to the organisms, particularly the plankton abundance, which serves as indicator of pollutants. This study is designed to assess the plankton abundance in relevance with anthropogenic activity. Understanding the relationship between plankton populations and anthropogenic activities that indicate pollution will help formulating effective strategies to protect and preserve aquatic biodiversity in the region. According to Kumala et al. 2013 and Sharma of 2018, the river system is one of the productive ecosystems of life on Earth and are major habitats of biodiversity, given that it hosts a wide variety of plants and animals including the plankton. They are very crucial for productivity. Plankton is a microscopic microorganism, free-floating creature in the water. There are two types of plankton, the phytoplankton, which is a plant-like organism, and the zooplankton, an animal-like. Global food webs would probably collapse in the absence of plankton according to McCullough of 2019. That's why their population is essential to aquatic ecosystems. Anthropogenic activities refer to all actions that are either caused by or connected to human influence that constantly affect the water habitat, including the presence of microorganisms which also affect itself. According to Hale et al. Of 2015, the occurrence of urbanization, industrialization, and population growth results in a rise in anthropogenic activities such as fertilization of agriculture, wastewater discharge, urban runoff, and nutrient input. These activities substantially limit the capacity of aquatic ecosystems to self-remediate. It is evident that human activities have a significant impact on the trophic status of aquatic ecosystems, specifically the plankton productivity. The problem statement of this study: anthropogenic activities play a vital ecosystem problem. This type of activity will affect water habitat and the plankton abundance, such as improper waste disposal, laundry activity, bath washing, agriculture activity, and so on. Nursery River is one of the water resources in Dinapigi, Isabela that are mostly visited and which they use to make fun, or they called it picnic, wherein during the activities they also use it for drinking. However, the majority of people living along the coast are unaware of how their actions affect the species that thrive in the river, particularly the plankton, which acts as a pollution indicator. The anthropogenic activities conducted is significant to the abundance of plankton in the nursery river as well as in human and ecosystem. The significance of this study: this study will provide valuable baseline data for future ecological monitoring and contribute to the sustainable management of the nursery river ecosystem. Understanding the relationship between plankton populations and anthropogenic activities that indicate pollution will help in formulating effective strategies to protect and preserve aquatic biodiversity in the region. The assessment aims to investigate the abundance and diversity of plankton in the nursery river Dinapigi, Isabela, and analyze their correlation with human activities. Specifically, aims to measure the abundance of phytoplankton and zooplankton in upstream and downstream of the Nursery River in Dinapic, Isabela. Identify the species composition of the plankton community to determine the presence of indicator species for pollution, and assess water quality parameters such as pH, dissolved oxygen, temperature, nutrient levels to understand the environmental conditions of the Nursery River in Dinapic, Isabela, to analyze the impact of the anthropogenic activities conducted and assess if there's occurrence of water pollution in Nursery River in Dinapic, Isabela, and to provide recommendations for mitigating pollution and preserving the Nursery River's ecological health in Dinapic, Isabela. This study mainly focused on assessing the abundance of plankton in the Nursery River in Dinapic, Isabela, and determine the relationship between plankton populations and anthropogenic activities in Nursery River in Dinapic, Isabela. The researcher will be used plankton net as method in collecting plankton and will be having two sample sites, the upstream and downstream, to evaluate human activities conducted within the river. So the duration of this study, once a month of assessment on plankton abundance in reference with anthropogenic activities will be conducted at Nursery River in Dinapic, Isabela. So the study will be conducted from October 2024 to April 2025. So the sampling site of this study, the study will be conducted in Nursery River in Dinapic, Isabela. The study will be having two stations, particularly in the upstream, which considered as the control site with minimal human impact, and downstream where near agricultural lands and residential areas potentially impacted by runoff and waste discharge. So sa sampling procedure, first is the evaluation of anthropogenic activities. So evaluation of conducted anthropogenic activities in the upstream and downstream of Nursery River in Dinapic, Isabela will be observed by the researcher, and survey questionnaires will be used and given to the citizens nearby the stations or the sampling sites. For the collection of plankton, each station sites will be collected 5 liters of water in mid-surface or 3-meter depth using a water sampler. The water will be filtered using plankton net with a mesh size of 20 micrometer for phytoplankton and 60 micrometer for zooplankton, and will be placed in a 100 ml polyethylene bottle. Then Lugol's iodine solution will be used as preservative. The identification will be conducted in Laboratory of Freshwater Fisheries Center at Etiaga, Isabela. Then the collected samples will be concentrated using centrifuge or settling method. Standard compound microscope will be used in identification and enumeration of plankton. Water sampling, water quality parameters will be measured using standard method. Parameters including the pH, dissolved oxygen, temperature, the nitrate, phosphate, and nutrient levels in relevance to pollution indicator. In identifying plankton biodiversity, the abundance of plankton will be estimated using the proportion of species in the figure 1 by Saiful Islam et al. Of 2022 and an algal pollution indices of Palmer 1969 based on genus in figure 2. So for the data analysis, the abundance and diversity of plankton will be calculated using Shannon-Weiner Diversity Index and Evenness. So the statistical analysis will be used Microsoft Excel. Pearson's correlation will be used to analyze the indirect relationship of plankton abundance and diversity with pollution indicators such as the nutrient levels and pollutant concentration. Expected results of this study, the assessment of plankton abundance in reference with anthropogenic activity in Nursery River in Dinapic, Isabela will be determined the abundance of phytoplankton and zooplankton in upstream and downstream of the Nursery River. So the species composition of plankton community and the presence of indicator species for pollution. The water quality parameters such as pH, dissolved oxygen, temperature, the nutrient levels to understand the environmental conditions of the Nursery River. So that's all, and thank you for listening.